Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullah. Today, inshallah, we are going to start our practical classes in physiology. And as I mentioned before, physiology is an experimental subject where the knowledge is derived from experimental evidence. Our practical classes today will be on the nerve and muscle. And this practical class is the first step in research. Because one of the mission of the university is to have a graduate who can solve problems in the future. والحل بتاع المشاكل هو عن طريق البحوث وعن طريق الريسيرش. So for any experiment, we consider experiment as a problem. For any experiment, there is a title. For any experiment, there is a title. So we start by looking at our title, the sciatic nerve and gastrocnemius muscle preparation. Today we are going to isolate the sciatic nerve and gastrocnemius muscle, and we are going to use our experimental animal is a frog. And as I tell you before also, the frog is an ideal experimental animal in physiology because the size of the frog is manageable, and also the life cycle of the frog is short, and it's available. Because the life cycle is short, their production is in a short period. So it's available at any time except during winter time. So this is why we are doing this recording. At least it helps during when the frog is not available. For any experiment, there is an objective. Our objective, we can know the objective from the title. Our objective is to isolate the sciatic nerve and gastrocnemius muscle. After we isolate our preparation, we are going to demonstrate simple muscle contraction. We are going to demonstrate simple muscle contraction or simple muscle twitch. For this experiment, to fulfill our objective and to do this experiment, we need material, and also we have to follow certain methods. One of the most important item in the material is the experimental animal. And in these experiments, we are using the frog as an experimental animal for the previous reason mentioned. Also, the frog tissue can tolerate experimental manipulation, and the tissue will be viable for a long time. To isolate the sciatic nerve and gastrocnemius muscle, we need to do dissection on the frog, and we cannot do the dissection unless we render the animal insensible to pain, and we do that by performing frog pithing. When we do the frog pithing, the frog is going to tolerate the manipulation. Frog pithing is going to tolerate the dissection procedure, because it will be painful without pithing. So after pithing, we start our dissection, and we use the dissection tools. But we perform pithing by inserting the seeker between the skull and the first cervical vertebrae. We insert the seeker or the needle to cut the spinal cord and to destroy the brain. By destroying the brain, it's considered as single pithing, but when we destroy the brain and the spinal cord, it is known as double pithing. The sign of successful pithing is a paralysis of the hind limbs of the frog. After that, we put the frog on the dissection board, and we start our dissection by making incision around the waist of the frog. And after making the incision, we remove the trouser of skin, the skin which is covering the hind limbs of the frog, to visualize the gastrocnemius muscle and the Achilles tendon. To further, we do further dissection to visualize also the sciatic nerve. And to see the sciatic nerve, we need to open the abdomen. We open the abdomen and we remove the visceral organ cranially, and we will see the two sciatic nerve emerging from the sacrum and the last lumbar vertebrae. We follow the sciatic nerve by dissection. After we cut the urostyle, when we cut the urostyle, it's easy to make longitudinal cut to have two preparation. We have to have two preparation of the hind legs. Usually after we remove the visceral organ cranially, we make our first cut above the first lumbar vertebrae, so that to see the side from where the sciatic nerve emerge.